Hello, I am Jonaki Sen and I am a member of the faculty in the Department of Biological Sciences and Bioengineering at IIT Kanpur. In my lab, we are interested in studying how the brain develops, and today I am going to tell you the results from a study in which we were actually looking at how the two cerebral hemispheres of our brain form. So, this study was recently published in the journal Development, and today I am going to tell you about how we actually found this out. So, if you imagine the brain in the embryo, it looks like a balloon, and this balloon looks in this shape, and the top of this balloon is known as the roof plate. So, this roof plate region undergoes a morphogenetic change in the form of a fold which happens like this, and this moves downwards and slowly it meets up with the bottom, and then the single vesicle separates into two chambers. These two chambers in future will become the hemispheres of the adult brain. So, now that you have understanding of how this process happens, I will tell you that we were interested in understanding the molecular mechanisms that might be involved in this roof plate invagination process. Now, why is this important? Because if this does not happen properly, this results in a developmental disorder which is pretty devastating, and it is known as holoprosencephaly. In the most severe form, this is of course lethal, but in the milder versions as well, there are several cognitive defects in such individuals. So, this study that we undertook was to understand what makes the roof plate invaginate in this manner, and to look at that we used the chicken embryo as a model organism. So, in this study we few years back we actually discovered that a vitamin A derived molecule called retinoic acid is very important for this invagination to happen. How did we know that? We blocked retinoic acid signaling in this roof plate region, and we found then this invagination does not take place and the roof plate remains flat, which looks very much like a holoprosencephalic brain. After knowing this, we were interested in understanding how does retinoic acid actually make this happen. So, to do that we had a serendipitous finding through another study where we were looking at the expression of metabolism related genes, and we found that the expression of one such gene known as CNKSR2 was located exactly in the middle of the roof plate where there was active retinoic acid signaling present. So, this was too much of a coincidence to be ignored. So, what we did is we wanted to see what happens to CNKSR2 if we disrupt retinoic acid signaling. So, when we blocked retinoic acid signaling in the midline, we found that indeed the CNKSR2 expression was lost. So, this indicated that CNKSR2's expression is dependent on retinoic acid signaling. Now, CNKSR2 stands for connector enhancer kinase suppressor of RAS2, and this is a modulator of RAS signaling. So, we also wanted to find out how does CNKSR2 actually modulate the process of forebrain midline invagination. So, to do that we then knocked down the expression of CNKSR2 using RNAi or RNA interference based method, and we indeed found that this also led to invagination defects. And after this we also found that it is a modulator of RAS-RAF signaling. So, it inhibits RAS-RAF signaling in the midline. So, everywhere you have high amounts of RAS-RAF signaling except in the very middle. What we did then next was to knock down the expression of CNKSR2 and at the same time inactivate RAS-RAF MEK signaling in this middle region. And lo and behold, this rescued the midline invagination defect, and now invagination was taking place almost normally. So, in conclusion, our study has found two novel players that are involved in the process of roof plate midline invagination. These are CNKSR2 and RAS-RAF MEK signaling, and this paper has been recently published in Development, and you can go to the website and check out the details there. Thank you.